Hello, everyone. Welcome to Wiley and welcome to our webinar today. The title of our webinar today is Beyond Aging, Revolutionary Research in Cellular Senescence and Regenerative Medicine. Our first speaker will be Dr. Professor Janosch Hildebrand. His presentation will be titled Latest Skin Biology Research Applications from Coburg University. And before we send it over to Dr. Janosch, I'd like to just remind everybody that they can make comments for the live Q&A after the presentations that we have for you today. So please find the chat box on your screen somewhere, and you can ask a question at any time during the presentations, and then we'll try to address them in the live Q&A. Also want to remind everybody that this webinar will be available on demand after it is over. You should get an email in your inbox giving you a link so you can watch it, or you can share it with someone else who you think might want to watch it. So without further ado, let's hand it over to Dr. Janosch Hildebrand and his first presentation. Thank you.
Where does aging begin and where does it end? My name is Janosch Hildebrand. I am a professor of biomedical analytics. I conduct research in the field of molecular aging. We use Memmert incubators to take the next steps in our research, as these provide a stable and controlled environment, which is essential for our experiments. And if we are able to influence the process of aging, we could potentially slow down, prevent, or even reverse age-related illnesses in the future. If we look at an entire lifespan and understand this as aging, then there are many influencing factors, for example our diet, UV radiation, or our genetic makeup. And we need to create reproducible environments in the laboratory in order to study individual aging variables in detail. We mainly use so-called omics methods to analyze genes, proteins, and metabolites. One area of research is the study of skin, because skin has a clear phenotype to begin with. That means I can see whether someone is old or young, and we are trying to understand what happens at a molecular level. For example, we take the cells, we culture them, we stress them, we allow them to age, and then we analyze whether these cells can still repair their own DNA properly.
Yeah, hello everybody. Welcome also from my side. My name is Janosch Hildebrand. I'm a professor for biomedical analysis at Coburg University, focusing on aging research and rejuvenation research, and especially skin biology. Today, I would like to provide an overview of the technology and research portfolio at my lab and our university, and show you how we leveraged our technologies to analyze data from the Human Protein Atlas to identify proteins involved in epidermal differentiation and keratinocyte biology. So at Coburg University, we have an end-to-end approach from analysis of cells and tissues to multi-omics in data analysis to finally gain biological insights. We have a strong background in cell culture technology, ranging from primary cell cultures to stem cells and three-dimensional organoids, especially human skin models. Beside all classical molecular biology methods, our major pillar, as you can see here under number 2, is our multi-omics pipeline. We use next-generation sequencing technologies for transcriptome analysis and mass spectrometry for proteomics using TIMS-TOF machines and QTOF machines for metabolome analysis and measurements. We have established workflows for cell culture samples, serum and tissue samples, as well as for exosomes from different matrices. The third pillar is our high-content screening facility, where we use phenotypic imaging for cellular analysis and screening approaches. Coupled with advanced image analysis of high-content imaging data, and in addition also immunohistochemistry data, this provides rich insights. All this comes with a lot of data analysis, and we have a very strong background in bioinformatics to gain real biological insights, because this is our major aim, from multi-omics data, also from bulk approaches, and also single cell approaches. So if you would like to work together with us in these fields, then I'm more than happy, and feel free to contact me afterwards. So today, I would now like to show some unpublished data from our lab, how we are currently analyzing skin data sets from the human protein atlas to identify in a systematic way proteins and genes involved in epidermal biology and keratinocyte differentiation. So the question is, why is this important? This is important because the epidermis forms the outer barrier of our skin, and to do so, the keratinocytes in the epidermis undergo a so-called terminal differentiation process. In the basal cell layer, as shown here on the left side, proliferative cells deliver new cell material to ensure the high epidermis turnover. To enable, on the other hand, epidermal barrier function, these cells leave the cell cycle from time to time and start a terminal differentiation process in the suprabasal cell layers. To understand the molecular processes behind this differentiation process on the one hand, and the regulation of the proliferative capacity, is of major interest to analyze skin aging processes, and also to develop skin aging treatments, and in addition also to develop treatments for atopic or psoriatic skin conditions, and in general analyze the function of the epidermal barrier. Our aim of the current project here is to answer now the central question: which proteins and genes are involved in and regulate keratinocyte differentiation? To answer this question and to get in vivo relevant insights, we developed a deep learning model to automatically analyze the epidermis in immunohistochemistry images from the human protein atlas skin datasets. The model was first of all trained on H&E images, and then a stepwise segmentation and transfer learning approach enabled at the end the analysis of immunohistochemistry stainings and images. The approach basically segments the epidermis and extract layer-wise the staining intensity for all proteins available in the human protein atlas skin dataset. Using statistical analysis and model fitting to the intensity data across the different layers of epidermis, we enable the identification of proteins which are differentially expressed across the entire human epidermis. To verify now the methodology, first of all, and our general approach, we first of all checked the results for known differentiation markers and epidermal stem cell markers. And as you can see above, there are typical differentiation markers like keratin 10, involucrin, or desmoglein 1, which were identified to be lowly expressed in basal cell layers, as you can see, and they were upregulated in suprabasal layers. On the other hand, the stem cell marker TP63, for example, is highly expressed in basal cell layers and decreased during the differentiation process. And the same is true for integrin alpha 6. You see here 3 different antibody stainings. Integrin alpha 6 is also a basal cell marker, and the same is true for the EGF receptor, for example, controlling the proliferation of basal cells in these basal cell layers. Taken together, this shows that the general approach and our methodology is able to identify differentially expressed proteins in human epidermis using immunohistochemistry stainings from the Human Protein Atlas skin dataset. After verifying the approach, we analyzed then the entire Human Protein Atlas skin dataset with our model and our approach. Therefore, we analyzed roughly 80,000 images corresponding to more than 13,000 proteins. After statistical analysis, we identified more than 2,000 proteins, which are differentially expressed across human epidermis. Out of this, more proteins, roughly 1,500, were downregulated and higher expressed in basal cell layers, and 500 roughly proteins were found to be higher expressed in suprabasal cell layers compared to basal undifferentiated cells. In automated literature research using LitSense revealed that the majority of these proteins were not so far reported to be involved in epidermis biology and or keratinocyte differentiation, indicating that our approach identified many new proteins regulating potentially keratinocyte differentiation in vivo. At the next step, we performed gene ontology enrichment analysis on the upregulated and downregulated protein sets. Interestingly, the enriched and overrepresented GO terms and processes identified reflect epidermal biology during keratinocyte differentiation. For example, terms associated with proliferation were overrepresented in the downregulated protein group, indicating that proteins expressed in basal cell layers regulate the proliferative capacity of human epidermis. Further terms associated with apoptosis, epigenetics, or signaling and immune response showed the same pattern. On the other hand, processes associated with terminal differentiation were overrepresented, especially in the group of upregulated proteins, as you can see above. This shows that the entire approach provides a novel resource for the systematic study of new proteins involved in keratinocyte differentiation. To now identify proteins which were regulated in vivo and were also accessible for in vitro validation and manipulation, we performed calcium-induced differentiation of primary human keratinocytes and analyzed the transcriptome using our NGS pipeline. The comparison of these transcriptome analyses to the immunohistochemistry data revealed that more than 600 proteins, so roughly 500 in the downregulated, you see it here on the left side, and roughly 160 for the upregulated ones on the right side, were differentially expressed during in vivo differentiation and showed the same direction of regulation within the transcriptome during calcium-induced differentiation in our transcriptome analysis. Currently, we analyze the identified transcription factors within these intersections, which were identified to be regulated in vivo and in vitro to understand the gene regulation networks in epidermis. So our data hopefully can provide a useful resource for the systematic study and identification of novel regulators of keratinocyte differentiation and, for example, can inform target selection for the cosmetic industry and pharmaceutical industry in the mentioned indications. At the end, I would like to say thank you to my great team and my colleagues, and thanks for your attention.
Thank you, Professor Dr. Hildebrand, for that interesting discussion. Our next presentation will be a technical discussion of the CO₂ incubator produced by Memmert that we just saw in the case study by Professor Hildebrand. Our speaker today will be Alexander Kowalchuk. He's a Senior Manager of Product Management at Memmert. And before I turn it over to Alexander, I'd just like to remind everybody to please use the comment box that you see on your screen. Once his presentation is over, we will have a live discussion with both of the presenters, and they'll answer all of your questions. So without further ado, let's get started, and I'll hand it over to Alexander Kowalchuk. Thank you.
Hello, everyone. My name is Alexander, and in this part of our webinar, I would like to take a closer look at the essential role of CO₂ incubation in cell culture, focusing on the key environmental conditions required for successful incubation. We will begin with an overview of product specifications, followed by some technical features, and you will also learn about available options and accessories designed to support a wide range of laboratory needs. Finally, we will take a closer look at applications that highlight the performance and reliability of CO₂ incubators in practice. In CO₂ incubators, carbon dioxide plays a key role in creating the optimal environment for cell and organism growth. One of the primary functions is regulation of pH level. When CO₂ dissolves in water, it forms carbonic acid, which helps maintain the pH of the growth medium based on a buffer solution within the ideal range. This balance is critical as many cell types require a stable pH to thrive and function properly. Heating system is a very important part of every CO₂ incubator, and precise temperature regulation is important. Otherwise, incubated cell culture stops growing or just may die in the worst case. To avoid any temperature overshoots, the incubator's advanced heating system ensures consistent and stable temperatures throughout the entire chamber. With direct six-sided heating as insulating jacket and heated inner glass door and seals, it delivers fast heat up and quick recovery after door openings. Designed for optimal growth conditions, it maintains the ideal breeding temperature of 37 degrees, equally to the human body temperature. But only temperature control is not enough for successful incubation. The other part of success is fast and precise control of relative humidity to avoid uncontrolled condensation and ensure optimal conditions for sensitive cultures. To prevent drying out, the humidity should ideally remain above 80%. This level can be achieved with a tray filled with water and located in the chamber. Together with this, the exact humidity value cannot be controlled. Let's see how humidification in CO₂ incubators is arranged. The simplest way to moisturize the chamber is water tray. This approach allows to create humidity up to 93% just by evaporation from the water tray. The process of evaporation and condensation is a cycle due to a Peltier cold trap at the back wall. By means of this cold trap, excessive moisture in the chamber is condensed and water flows back through a rubber leap, which you can see on this slide. And this rubber leap is a black part of a water tray. Simple, but not always enough. Can we achieve high humidity values? And if yes, how? Let's move on and look. The solution for maintaining high humidity range is active humidification. With digital humidity control, option K7, both humidity rise time and recovery time after door openings are significantly faster compared to passive systems. This process is provided by a steam generator and introducing water vapor directly into the chamber. Precise regulation is ensured through continuous monitoring and feedback from the integrated capacitive humidity sensor. Interior dehumidification is efficiently managed by a controlled supply of fresh air through a sterile filter, maintaining optimal conditions at all times. Let's take a look at the product specification of CO₂ incubators. Memmert ICO incubators are available in 4 sizes: 56, 107, 156, and 241 liters, with a setting optimal temperature range from +18 to +50 degrees. But together with this, working range is at least 5 degrees above ambient temperature, up to +50. There is no humidity control with a passive humidification provided by means of water tray located in the chamber. In this case, humidity in the chamber will be kept at maximum of 93%. But for precise humidity control from 40 to 97%, the incubator can be optionally equipped with option K7 and active humidification and dehumidification systems with a setting accuracy of 0.5%. It goes without saying that every unit has CO₂ control in the range from 0 to 20%. The standard incubator comes fully equipped for reliable cell cultivation, featuring stainless steel shelves and water tray, a CO₂ connection set, and a high temperature sterilization program at +180 degrees for 60 minutes, including all internal components and individually selectable parameters such as CO₂ level, optional control of oxygen, and active humidity control for maximum flexibility. Every device is available as a medical device, which is very important for medical applications if we talk about in vitro fertilization or other medical purposes. As for applications, CO₂ incubators play an essential role for a wide range of applications, including bioassays, tissue research, and cell cultivation in stem cell studies. They support gene expression experiments, in vitro fertilization, and embryo storage. With optional O2 module, they can be also an ideal solution for anaerobic breathing. Additionally, they play a key role in quality assurance of drinking water. To sum up all the benefits of Memmert ICO incubators, we should highlight that ICO offer several advantages that make them stand out in the market. Built with a durable stainless steel, they provide a long-lasting robustness and are easy to maintain and, what's more important, to clean. 6-size heating system, including heated glass door, ensures uniform temperature distribution, creating the ideal environment for cell cultures. Designed with the user in mind, the incubators feature user-friendly operating concept that simplifies control and monitoring. In addition, all incubators are equipped with advanced atmo control software and offer precise regulation of all parameters. Additionally, every incubator includes a comprehensive sterilization function that sterilizes all internal components and sensor inclusively at 180 degrees Celsius for 60 minutes, ensuring a contamination-free workspace. To top of it, the incubators come with a 3-year warranty and a calibration certificate, giving users peace of mind with a reliable performance and verified accuracy. I would like to say a couple of words about our company, Memmert. Memmert is a globally recognized manufacturer of laboratory equipment, specializing in climate control and temperature regulation solutions, combining cutting-edge technologies with precise engineering to deliver reliable products tailored to meet the diverse needs of scientific research, industry, and healthcare. The outstanding level of performance are not achieved by chance, but they are a result of the dedication of the team of more than 500 employees. Each member of this team contributes their expertise and passion, working together to develop and manufacture products that consistently meet customer expectations. The collective commitment ensures that Memmert remains at the forefront of technology, delivering solutions that support critical scientific research and industrial processes worldwide. Now we have 2 production facilities in Germany located in Schwabach and Büchenbach, and also sales representative companies in several countries such as China, India, Central European office in Bratislava, and of course the subsidiary office in the United States. All the products are made in Germany, with manufacturing taking place, as mentioned, at 2 production facilities located in Schwabach and Büchenbach, and these modern plants allow Memmert to produce appliances efficiently while maintaining the high standards of quality and precision. Beyond efficiency, the company places great emphasis on environmental responsibility, ensuring that all manufacturing processes are conducted in eco-friendly manner. It goes without saying that the highest production standards are the priority for Memmert. And with a recently installed production line allowing for a highly economical and efficient manufacturing, we may ensure that the quality of products remains stable and outstanding. The advanced setup minimizes waste and maximizes overall performance, ensuring we deliver top quality products sustainably. Each Memmert device undergoes individual performance and accuracy testing in our efficient test field. Every chamber is carefully calibrated and comes with its own calibration certificate. Additionally, all Memmert products comply with the latest DIN guideline, which involve stricter testing for temperature, homogeneity, and reliable operations. But not only CO₂ incubators, the product range of Memmert consists of universal ovens, incubators, Peltier-cooled incubators, compressor devices such as incubators and climate chambers, and also water bath. And in any case, any user can find a suitable solution for every application. Thank you for the attention, and I'm passing the word back to other participants.
Okay, so the first question I'd like to ask is, and please excuse if my internet drops off again, maybe we could just have a brief discussion of the equipment that you use in your lab, Janosch or Dr. Hildebrand, not from a technical perspective, but maybe how you use equipment. But before you answer that question, maybe Alex just could answer one question about the different machines. So the functionality of each of the sizes is exactly the same. Is that correct, Alex?
Yes, Tom, that is absolutely correct. It does matter which size you choose. The functionality can be adjusted to your needs with different options or with the standard models, but it doesn't depend on the size of the machine.
Okay, so the only difference is basically is not necessarily in the functionality, but just in the volume of samples that you can actually put inside the machinery.
Absolutely right.
Okay. And Dr. Hildebrand, maybe you could just briefly describe some of the uses that you use the machines for.
Yes, sure. So we use the ICO incubators for normal cell culture experiments, but also for more advanced techniques where we want to induce hypoxic environments, for example, to study hypoxia during skin aging and also keratinocyte biology mainly. This is one thing. And the other thing which can be done with these incubators is regulating the temperature profiles. And this could be informative, especially when we talk about circadian rhythms and the entrainment of cells in the cell culture environment.
Okay, we have a question from the audience. What is the role of hypoxia, HIF, and cellular senescence / aging?
Yes, so maybe I can share some thoughts about it. Yes, of course, hypoxia is important for aging, and there are many publications. And the field is still in discussions about the importance of hypoxic environments for aging. But for the skin, for example, it could be an important parameter, as, for example, the blood vessels decrease during aging. And so, especially in the epidermis, there could be in hypoxic environment in addition. Regarding the transcription factor, or the main factor HIF1 alpha, there it was shown in mice models or mouse models, for example, that knockout and knockdown of this gene leads to skin atrophy, for example, and impacted also the proliferation of keratinocytes, and especially in human skin models also.
Okay. Which role does sorry for leaning forward here, but these words are small. Which role does telomere preservation play in skin aging? Could you comment on this point?
Yeah, so this is a field of current discussions, to be honest. So there are publications showing it is important, other pointing that it's not so important. Of course, telomere stability and genome stability is an important factor for aging, for sure, and also for skin aging, especially as the keratinocytes have real high proliferation and turnover, basically, over the entire lifespan. So it could be an important factor for aging in addition, yes.
Okay. And Alexander, I think this is one for you. Is there a tri-gas incubator in Memmert portfolio to create a hypoxic environment?
This question is related directly to the first one to Dr. Hildebrand, because tri-gas incubators are used to create a hypoxic environment, and they're specifically engineered to regulate not only carbon dioxide level, but also oxygen and nitrogen levels. And in contrast to conventional CO₂ incubators, tri-gas units can create a low oxygen hypoxic environment, or even custom gas environments, which are crucial for some studies. But as for our incubators, the standard model includes only CO₂ inlet and control. However, with an optional upgrade that introduces a nitrogen inlet and oxygen control within the range from 1 to 20%, which exactly provides the required hypoxic conditions. And yes, as mentioned, these types of environment and these incubators are widely used in the fields such as stem cell research, oncology, especially IVF and any other types of tissue engineering.
Okay, and I think this is another question for you, Alex, as well. How can performance and recovery times of gas concentration and humidity level be improved? For a number of applications, frequent loading and unloading of samples and opening the door may result in a significant deviation, which can be a problem for the viability and quality of the cell culture.
That is also a good question, and that's great that we have this live session to discuss these issues, not only advantages, but also issues in some studies. To minimize deviations in gas concentrations and humidity levels, especially in applications where frequent door openings and sample handling occur, it's crucial to use an incubator that offers rapid recovery and precise environmental control. And as I mentioned in my presentation, the effective solution is implementing the optional humidity control, which allows to accurately regulate the humidity level even up to 97%. This helps maintain stable conditions even during repeated loading and unloading procedures. And additionally, the system can actively reduce humidity down to the level of 40%, which is not required in many cases for with a cell cultivation, but can be required for a number of other studies. I hope this explains how we can solve this problem.
Okay. This one is for you, Dr. Hildebrand. How can the data be used to inform skin aging research? And I'm assuming that they're talking about the data that you have generated or used in your labs.
Yes, the data we generated is basically a novel, large, and let's say genome-wide resource for expression patterns across human epidermis. And this is important for keratinocyte biology and also especially important for skin biology and also skin aging. So our strategy currently is to combine our analysis with large cohort expression data across the entire aging. And our aim is to identify genes or proteins which are deregulated during aging and also were found in our analysis to be differentially expressed in human epidermis, because this could be important, and these genes or proteins can be targets which are deregulated during the aging process and thereby disrupting the normal process of epidermal barrier function.
Okay. Are you planning, Doctor, are you planning to publish your findings with all the regulators you have identified?
Yes, we plan to publish at least some part of this data for sure, and eventually also the entire data set. Currently, we are working also together with some industry partners, and but this data and this project will be published soon, hopefully. Keep fingers crossed for it. And yes, then the entire data is also available, especially also about the regulators and the transcription factors, for example.
Okay. And have you looked at the effect of light on your research or the work that you're doing?
Yes, so I'm not sure what you mean with light, but maybe you mean UV light. And this is really, really important, of course, for human skin and especially for human epidermis.
If I might interrupt, Dr. Hildebrand, there's another question about UV, and maybe you could get both of them at the same time since you mentioned UV light. So have you looked at the effect of light is the first question. The second one is, how important is UV protection for skin aging, and what about retinoids? So maybe you can capture both of those in the same.
Yes, so UV protection is key for skin aging, for sure. And also for skin biology and keratinocyte differentiation, and especially DNA damage protection, for example. And what we found also in our data set here is that many proteins which are involved in DNA damage response are differentially expressed across the human epidermis, for example. So UV protection is key for the understanding of skin aging, and is a suitable target to increase the DNA damage response to counteract UV light-induced DNA damages.
Okay. What senescence-inducing factors are best suitable for keratinocytes?
Yes, there are multiple discussed factors. Sometimes you see, for example, P16 as a traditional one. You can use metabolic markers, for example, like beta-galactosidase. Stuff like this is also used for keratinocytes and is also useful. Our data also can be used to identify exactly such factors in future. When we publish our data, you will see many other factors which are maybe also involved in regulating the senescence of keratinocytes during aging.
Okay, and Dr. Hildebrand, does Coburg University have direct cooperation in aging research with some universities in the Balkan countries or in Central and Eastern Europe?
No, currently not in these regions. We have some cooperations, of course, with other researchers across Germany, for example, and Switzerland. in the field mainly of metabolomic studies, which we are running at our lab, and studying the effect of metabolism on skin aging.
Okay. How can your analysis inform target selection in the industry, as you mentioned on your slides?
Yes, I think our data is useful also for target selection and validation, because if you identify, for example, a target for in keratinocytes, which is differentially expressed, you want to know whether it's differentially expressed also in vivo and maybe regulating the epidermal biology. And so therefore, our data can be used to inform such target selection and check for expression in human skin, and then came up with hypothesis whether this gene is really a good target and also expressed in vivo. Because if you use in vivo keratinocytes, and this is also what our data shows so far, in vivo, there is a totally different situation. So other proteins are expressed which are not expressed, at least at the transcriptome level, in our keratinocytes in vitro. And so the in vivo situation can be different from the in vitro situation, and therefore our data can be a useful resource to compare to the in vivo situation afterwards.
Okay, and how do you plan to validate your insights in cell culture experiments?
Yes, we are currently running siRNA experiments where we systematically knock down specific and identified proteins. Thereby we focusing, as mentioned in the talk already, especially on novel transcription factors which were identified in our analysis. And we want to see whether these knockdown or siRNA transfections at the end leads to deregulated keratinocyte differentiation or proliferation.
Okay. Can your data be used to analyze basal cell carcinomas in addition to the other experiments that you're working on?
Yes, this is exactly the same as for skin aging research and also for the target selection. We are, a student of my lab is currently using our data and try to combine this data with publicly available expression data from basal cell carcinomas. And thereby you can search for opposite regulations. So for example, check for genes which are highly expressed during the differentiation process and which are downregulated during the formation of basal cell carcinomas. And this could be interesting because thereby we may identify new regulators of the proliferation switch during the formation of basal cell carcinomas.
Okay, I have one more question for Dr. Hildebrand, and then Alexander, I think I have one more question for you as well. Please, audience, don't forget, if you have a question, you can enter it in the chat box and we'll get to it. We still have about 15 or 14 minutes left. So Dr. Hildebrand, can you give more details about the biological function identified by your approach?
Yes, so the data is not published, so I cannot provide so many real details. But what I can tell is that we identified roughly 80 novel transcription factors which are differentially expressed during keratinocyte differentiation in vivo. And many of them are also regulated in vitro. And this is really interesting for us to analyze the gene networks controlling differentiation processes in skin and in the epidermis, and also especially the stem cell compartment and the epidermis stem cells itself. In this field, it is interesting that we found novel differentially expressed proteins which eventually regulate the TGF-beta signaling pathway, which could explain some of our data regarding the proliferative capacity of human keratinocytes in the epidermis.
Okay, Alex, just curious about the future of the machinery that you're working on at Memmert. What are the innovations that are upcoming or that people are working on in your industry that you're working on in particular? Does AI play a role? And if these machines are connected to any kind of network, is there security issues with the machinery as well?
Also a good question. We permanently are collecting the data, the input from the market and from the end users to check what we can improve and what can we integrate for further developments and further generation of equipment. And yes, of course, it goes without saying that we have an application, a software to monitor, to record all the data from all our machines. And also, we can offer even a mobile version for this. And yes, that's important to provide the respective level of security for this data. And that's obviously achieved in our new developments and last improvements in terms of our desktop software.
Okay, can you talk a little bit about the state-of-the-art ICO that is coming up early next year, fully connected to the lab and with full security?
That's true. We have a new development in progress, and actually that will be something different from what we have now. Even better in terms of controlling unit, and even better in terms of lab footprint and controlling the device, especially in a remote way, which is achieved in our current models, but it definitely can be improved and it will be improved to provide the remote access for the device from any point of the world. I would say this.
Okay. One more question for you from the audience, Dr. Hildebrand. Have you looked into the epigenetic differences underlying skin cell differentiation?
So my lab did not look so far in epigenetic regulations during skin or keratinocyte differentiation, but our data, especially this analysis, points to the regulation of epigenetic factors during keratinocyte differentiation. So as I showed in the talk, there's one slide with our gene ontology enrichment data, the overview of it. And there you can see that epigenetics and epigenetic factors and genes involved in epigenetic regulation are highly enriched in the proportion of genes or proteins which are mainly expressed in basal cell layers, pointing to the fact that these epigenetic changes mainly occur or are regulated in this compartment during the differentiation process.
Okay, and I'm going to throw that question that I just asked Alex to you, Dr. Hildebrand. What about your future and your future of research? Can you describe a little bit of what you're looking forward to in the near future, say the next year or so?
Yes, my lab is basically running research in skin aging and also skin rejuvenation or rejuvenation processes. And therefore, this analysis, what I showed today here, is an important resource for us to look in identified genes and proteins from other projects. So for example, we try, as I mentioned already, to combine this data with large cohort expression data across the chronological aging during skin aging. This is one main route currently. And we are also highly interested in the regulation of metabolism, especially where my lab is working since years on it, to understand how the metabolism change during aging and also during UV light irradiation, for example, as mentioned before. And in the field of rejuvenation, we started currently novel projects where we utilize the expression of Yamanaka factors, for example, to induce rejuvenation of skin, and also to leverage the concept of parabiosis for in vitro rejuvenation of keratinocytes and fibroblasts.
Okay. Well, we're not out of time yet, but we don't really have any more questions. So I'm going to end the webinar now. That was super interesting. Thanks to the audience for all the questions and for your interesting answers. Please don't forget that you will receive an email with a link that you can watch this over, or you can share with your friends who might want to watch it for the first time. And if we didn't get to any questions, if I missed them in the chat, sometimes it gets a little bit confusing. We'll send you an email and try to get your question answered through the email. And we have your contact information. So anyway, without further ado, I'm just going to end the webinar here and just say once again, thank you to Dr. Hildebrand and Alex Kowalczuk and Memmert for sponsoring this webinar. And hopefully we can see you at the next one. Thanks again and take care.